I'd like to now welcome our speaker. Bruno Kopersky is currently the head of IVF at iGenomics Brazil. He has worked more than 9 years in the field of reproductive genetics. In his previous experience, he has worked in major laboratories in Brazil. Bruno has experience in pre implantation genetic diagnosis, PGD, PGT-M, and PGT-SR, and pre-implantation genetic screening, PGS, PGTA, carrier screening, DNA sequencing, and molecular biology. He participated in the implementation of different laboratories in Brazil, United States, Mexico, and Spain. Bruno, you may now begin your presentation.
Hello everyone, thank you for the introduction. So my name is Bruno Kopreski and currently I'm the head of IVF here at iGenomics in Brazil. And today we are going to talk about the expanded carrier screening clinical research with ion Torrent CarrierSeq ECS. First of all, we have to talk about the disclaimers. The Thermo Fisher Scientific and its affiliates are not endorsing, recommending, or promoting any use of application of Thermo Fisher Scientific products presented by third parties during this seminar. Information and materials presented or provided by third parties are provided as in and without warranty of any kind, including regarding intellectual properties rights and reported results. Party presenting image, text, and material represents they have the right to do so. So today we're going to have three learning objectives. So the first one will be, what is a carrier screening and how is it used? The second of it will be expectation for NGS-based carrier screening clinical research, and the last but not least, the best practice for implementation and validation of the CarrierSeq ECS. So let's get started. What is the carrier screening and how we can use it? How is it used? The first thing that we have to know is what is a genetic disorder. So this is so important for us. So a genetic disorder is a disease caused in whole or in part by a change in the DNA sequencing away from the normal sequencing. So we call it a change in the DNA sequence when we have a change in a pair base, when we have a change from A to T, or say for instance when we have a deletion of 5 or 10 pair bases. This is all we can call a change in the DNA sequence. The genetic disorders can be caused by those 5 types of different parts of genetic mutations. The first of one, we call it a monogenic or single gene disorder. This is the disease that we have one mutation in only one gene, and we have, unfortunately, a disease. The second one is when we have the multi-factor inheritance disorder. So we must have mutation in multiple genes, or we can call it polygenic, to unfortunately have the disease. The third genetic disorder, we can call it when we have the combination of genetic mutations and the environmental factors. Say, for instance, when we have the autism, that we have gene mutations or genetic mutations, and unfortunately also the environmental factors to unfortunately have the disease. Of course, we also have alterations in the mitochondrial DNA that we can call it the mitochondrial disorder. And this is so important because the mitochondrial disorder, we have only, we can only have this disease when it's passed by our mothers, because we have all the mitochondria that we have in our body, we receive it from our mothers. And of course, when we have a chromosomal disorder, that we have in a change in the number or the structure of entire chromosome. The chromosome is the structure that carries all of our genes. Say, for instance, the chromosomal disorder that we talk, we can call it the Down syndrome. That is, unfortunately, when we have three copies of the chromosome 21. So, to continue, what is the single gene disorder? We call it a single gene disorder. Is a hereditary disorder that is caused by a presence of a mutation or mutations in a particular gene. These diseases are rare individually, but according to the data from the World Health Organization, the global prevalence of this illness is 10 in 1,000 newborn infants. There are estimated that, taken together, indicate that this illness represents 20% of the causes of infantility mortality in developing countries, and that they are behind 18% of the intervention in pediatric hospitals. This is why it's so important to screen uh, all these types of mutations when we are thinking about to have pregnancy. So how can we classify the single gene disorders? So we have three different types of classification for this single gene disorders. So of course we can have this difference according to their inheritance pattern. So the first one, we call it a dominant disease. Those diseases involve the damage or a mutation in only one gene copy. So, as we can see in this image, when a person does not have the condition, we call it a non-carrier. But unfortunately, when a person has a mutation in this type of gene, we can call it affected. We just need only one mutation in only one gene copy to have the condition. We have to be aware that, of course, we do have two gene copies for every single gene that we have. The second classification that we have for single gene disorders, we call it recessive disease. That include or occur due to the damage in both copy of the gene. So, as we can see it, a person that does not have the condition, we call it non-carrier. But if a person has just one mutation in just one gene copy, we call it a carrier. But if a person receives a mutation from his father and his mother, this person will have two mutations in the two gene copies and unfortunately has the condition and we call it affected. And the third category that we classify the single gene disorders, we call it X-linked diseases. That are monogenic disorders that are linked to defective genes on the X chromosome. And this is important to tell you that all the men has one X chromosome and one Y chromosome, and all the women have two X chromosomes normally. So if we talk that a man does not have the condition, in this X chromosome, we don't have any mutation. But if we can talk that a woman is a carrier for that disease, it means that in one of the chromosomes X in this gene, she has a mutation. And unfortunately, it can pass by the descendants. If unfortunately this woman that we call it a carrier pass on the X mutated to a to a male child, unfortunately, this male will have the disease because it only has one X. So, what we can call about the frequency for this single gene disorder? We can see it right now in our literature that we have that 82% of individuals are carrier for at least one genetic condition and have a chance of passing it to their children. Even if their cells are healthy, every person has an average of two genetic mutations. And unfortunately, 5% of couples carries the same mutation. And there are a huge problem to screen it and to test it, those persons to check if unfortunately they do carry the same mutation or mutations in the same gene, and unfortunately their children can have a 25% to become affected. Of course, we are talking about the recessive disorders. Most carriers of genetic mutation don't have a family history of this disorder. And this is so important. So, because generally, parents only realize they are carriers of a serious genetic disorders after, unfortunately, an affected child is born. For these reasons, if both parents carry a mutation in the same gene, they'll be at much higher risk of having an affected child. And we can talk this risk, it will be 25%. This is why it is so important to think about the carrier screening in those type of patients. In this slide, we have we can see that the improved knowledge on the interpretation of genetic variations in our genome, it is so important for all the testing that we are going to perform it today and from 5 or 10 years from now. Of course, we all know that we live in a multi-ethnic society. It is so important to have this different uh, this difference in genetics that we have in different ethnicities. It is so important for us to realize what is our our genome like as a society. In this graphic, we can see that the first genome that sequenced was sequenced in 2001 when it was finished the Human Genome Project, and to to sequence one genome, complete genome, it took days or maybe weeks or years to perform it. Right now, with all the updates that we have in our sequencers, in our bioinformatics, and everything that we have right now, we can sequence a whole genome in less than 48 hours. This is so important for us to be able to provide this type of test for every single one that we have in our population. So, what are the recommendations for the expanded carrier screening gene panel development? And this is so important because the expanded carrier screening is an acceptable strategy for pregnancy and prenatal carrier screening, provided that specific criteria for the testing are met. And this is so important that this is not Bruno saying, this is the ACOG or the SMG talking about the statements and positions regarding the expanded carrier screen. So, all parents who are considering pregnancy or are already pregnant, regardless of the screening strategy and ethnicity, should be offered the carrier screening for cystic fibrosis and spinal muscular atrophy, as well as a complete body count and screening for thalassemia and hemoglobinopathies. Fragile X pre-mutation carrier screening is recommended for women with a family history of Fragile X-related disorders or intellectual disability suggestive of Fragile X syndrome, or women with a personal history of ovarian insufficiency. Additional screening also may be indicated based on the family history or specific ethnicity. Additionally, screening conditions should be able to diagnose prenatally and make effort opportunity for antenatal intervention to improve perinatal outcomes, change to the delivery management to optimize newborn and infant outcomes, and education of their parents about special care needs after birth. And finally, expanded carrier screening panels may include rare conditions. For such disorders, the precise carrier frequency as well as the proportion of the condition causing variants that can be detected may be unknown. Therefore, the calculation of residual risk after negative screening tests may not be possible for all conditions. And this is so important that for everyone that is listening to and is thinking about the carrier screening, that expanded carrier screening should not include autosomal dominant or adult onset multifactor conditions. So, when we think about expanded carrier screening, or when we think about carrier screening, the test is recommended in the following case. For a couple that wants to form a family, to know the risk of transmitting hereditary disorders to their child before an assisted reproductive treatment. This is so important. Right now, we can see that in the United States, uh, around like more than the majority of persons that are performing an assisted reproductive treatment are thinking about the carrier screening because of this importance. And of course, it's important to perform a carrier screening before a treatment with donor sperm or eggs to realize if unfortunately this donor uh, has a condition that maybe a person has it also. Here, I wanted to provide you the Brazilian scenario that we have for carrier screening. Unfortunately, the carrier screening here in Brazil, it's not developed as we have in the United States, for example. Uh, right now, we don't have the majority of persons that are thinking about the assisted reproductive technicians uh, looking for this type of test. So, to for you to realize the estimated carrier frequency for sick cell trait in Brazil, it's 1 in 25 in the general population, and it is 1 in 10 in the Afro-descendants. Here in Brazil, we have the ninth largest Ashkenazi Jewish population in the world. And unfortunately, the Ashkenazi Jew individuals are at risk to be carriers for certain autosomal recessive conditions, such as Tay-Sachs disease. The carrier frequency for cystic fibrosis is increased in the individuals of European descent compared to other ancestral groups. This is so important when we talk about the populations in Europe or the populations in the United States. Uh, in the populations of European descent, the carrier frequency is 1 in 25. European origins make up approximately 60% of the general Brazilian population, and as high as 78% of the population in the southern region, which means that many Brazilians are increased risk for being carried for cystic fibrosis, for example. And we have this image that I looked out in an article that we have here in Brazil that was published in the literature that the research has asked to 353 persons if this person have ever heard about some disorders. And by unfortunately, we have an unknown knowledge of a lot of genetic diseases. Say, for instance, we have like around just 20% of knowledge of what is Tay-Sachs, or maybe 20 to 25% of persons that knows what is thalassemia. The cystic fibrosis is a disease that the majority of persons here in Brazil knows at around 70%, but it's not 100% of the Brazilians that knows that we have a disease that is called a cystic fibrosis, and this is a disease that unfortunately is caused by a genetic mutation. So this is our part to become presenters of this knowledge or this information to raise awareness to all the Brazilians that we have here, or of course all the people in the world. In this other publication that we have, this was a publication published in 2021 by Caio Buaco. It's a doctor here in Brazil that I'm acquainted with, and a huge researcher. He has provided us by analyzing samples that were sequenced to check to see the test of a carrier screening. So a total of 425 occurrences of 351 different variants was found in a Brazilian population, and 47 variants were recruited more than once. And this is so important. Were reported in 278 different genes for 230 patients, a percentage of 71.9% in a cohort of 320 patients. For 90 cases, or 28.1%, no variants were reported. And in the overall, they observed an average of 1.3.2 reported variants per patient. And this is so important for us because right now we have this, unfortunately, this this knowledge that we don't have any variants, we don't have any genetic mutation. But we can see in this publication that more than 70% of the patients have a mutation. Of course, this mutation does not carry or does not become a disease. We are not talking about that 70% of the persons are genetic with a genetic disease, but there 70% of this population there that carries a mutation and can be at risk if they choose or unfortunately if their partners has the same mutation or in the same genes. So this is why it's so important to test the population regarding the carrier screening when we're talking about genetic population, or to think about all the diseases that we can not to this, those diseases that we can not pass on to our children. So this is why it is so important. This is the second part of this presentation when we're going to talk about the expectations for the NGS-based carrier screening in clinical research. When we talk about the NGS-based carrier screening, we have to talk about the advances and the decreasing cost of the NGS enables the carrier screening research across a broader range of disorders using the expanded carrier screening gene panels. The next generation sequencing based expanded carrier screening research has demonstrated the identification more at risk pregnancy in comparison to carrier screening research based on ethnicity and family history. In my opinion, with my experience, we are able to see when we perform and we compare the ion torrent carrier screening, that expanded carrier screening, to other panels that we used to perform, we are able to see a more complete analysis of those types of genes. We have uh, more than 400 genes. We have 420 genes that we are able to see the results. And for us, it's a really good opportunity to sequence those types of genes and to have and to give a result to our samples in a more broad example. And for us, when we started the validation and we started to perform the analysis with those kits, we have a really good results analyzing such for such this amount of genes. So the panel designs demonstrate an excellent coverage that results only 33 genes with a single exon level resolution. We have more than 36,000 SNVs in Dells and SNVs. And it is really good for us because we had an expectation when we started the test that maybe the test won't be able to cover all the SNVs that we were looking for. But by the contrary, we had a really good opportunity to have in just one kit the opportunity to analyze the genes, the SNVs, the indels, and the CNVs. Uh, when we're talking about these types of tests, of course, we do have special cases genes, and it is we have a lot of genes that we must have an extra effort to increase the coverage, and it was the most challenging part when we started the validation with this panel because of course we have a multidisciplinary person that works at the laboratory, and every single one has an opinion about which type of NGS we are going to perform and everything. And of course we do have a fear that won't be able to see some mutations or some variants in those types of challenging genes. Uh, in this panel, it was really good because this effort to increase the amplicon coverage to enable potential single exon in these 33 focus genes, and take for instance the SFTR, the cystic fibrosis gene, and then we know that it's a really difficult gene to sequence. We had a really good opinion and we had a really good experience in this panel. We are able to see in our validation that the gene was covered really well. All the samples that we have that unfortunately or have any type of variant in this gene, we are able to see the result when we analyze in the pipeline. Of course, we do sequence all the samples that we perform in our validation with the S5 sequencer, and we had a really good opportunity to see when we're looking at the carrier report, that is a software designed by Thermo Scientific, that we were able to see an end-to-end type of kit, because we received every single part of the kit that we are able to to have the library, to prepare the library, to sequencing, and of course we do have the bioinformatics pipeline that helps us to to find out all the variants that we are looking for. So in those slides, we are talking about the validation that Thermo Scientific performed, and we are able to see the SNVs and indel colon. We have an accuracy of 99.5, the sensitivity about 100%, and the specificity around 99.5%. And in our validation, in our own validation, we also see results according to the same types of thermal has found in this validation. The CNV columns, we have a accuracy of 99.46%. Also, what's good because we also in our validation we also sequence. We already knew that those samples were carrier positive samples, and we have around the same sensitivity and specificity that Thermo has in this validation. We have also in our validation around 99% in sensitivity and specificity when we are talking about those positive, those carrier positive samples. The SNVs was also another challenging part of this type of analysis because we all know that the SNV columns in when we sequence it, we have some technical difficulties, but we do have a really good concordancy when we perform the test with this panel. We have Thermo has in this validation a concordancy of 100%. Uh, as we said before, the panel included technically challenging genes associated with several relevant genes. And it was, my opinion, the most challenging part of this validation or this panel. Because we all know that, for example, HPA1 and HPA2 are really good, really difficult genes to sequence. We all know the SM1, that is a gene that unfortunately causes spinal muscle atrophy. That is a really difficult gene to see the variant call and to see the gene carrier status. And we can see it here in in this data that was provided by Thermo that we have a sensibility of 100%. And in our validation, it was a really concerning part of it, and we analyzed some known SMN gene carrier status, and we do have a really high sensitivity for this type of genes. Uh, they have achieved 100% sensitivity and specificity for HP1 and HP2 in the variant calling and 100% of concordancy in the GBA variant calling. We do have the same types of data and the same types of rate in our own validation, and this is why we started to perform the analysis with this type of panel. It is for us, it was a really good opportunity to see to maximize all the possibilities that we have when we sequencing those types of samples, when we use the NGS to analyze those types of positive samples that we had. So to talk about a little bit in this validation, it is so important because thermal and also when we perform our validation, we also use some Coriell samples. Uh, Thermo used a 23 Coriell samples library, and as we all know, the NA12878, one of the genome's most famous genomes, and every single one that works at laboratory knows that genome and knows every single variant that has in this genome. Thermo also use it, and we also use it when we are performing our validation, and we can see that this kit has more than 99% in intra and inter assay reproducibility, and it is really good when we are talking about those type of samples. Uh, we can see here in this slide that we have a concordancy more than 99.7% of SNVs and indel genotyping calls when we are using the NA12878 sample. In this validation, two operators each made a single library of this cell line that was sequencing in duplicate for a total of four tests. which showed a high analytical sensitivity, specificity, and accuracy across operator and library. The third part of this presentation, what are the best practices for the implementation and the validation of the CarrierSeq expanded carrier screen? So we have key steps in the analytical validation for this type of panel. And right now I'm going to divide it in three parts: the test development optimization, the test validation, and of course, one of the most important parts, the quality management. So, what are the activities when we talk about the test development optimization? So, we must define the test definition and the acceptable sample types. We must develop the variant calling and the annotation pipeline. Conduct the test performance assessment based on the test definition. And develop a variant confirmation strategy based on test performance. And at least assess the reference material and the positive controls for validation. And what are the outcomes for this part of this analytical validation? So we have the test definition with variant class reported established. We have the analytical pipeline established. We have the performance metrics defined, the test limitations identified with confirmation strategy developed, and the sample types and number of samples that was required or is required for a validation to determine a validation. So when we think about the test validation, we must have those types of activities. We must measure the test performance and accuracy with respect to variant class, variant size, and the genomic context. We must measure reproducibility, repeatability, and limits of detection. And, of course, use of laboratory positive controls for variant class beyond the SNVs and indel. In this part of the analytical validation, we must have the outcome of a documentation of every part of this test validation. The test accuracy it is established for all reportable variant classes. We have the reportable regions and the test limitations documented. We have the performance metrics for quality management plan definite. And the last part but not least, the quality management for this analytical validation. We have to define metrics for quality control monitoring of the test performance. Periodically sequencing of positive controls, and it is so important to see the reproducibility of your test, of your kit. The quality assurance monitoring of the test performance over time, and it's so important to always keep an eye on monitoring the test, monitoring the types of variants that you are looking in those types of panels. You have the outcome of a quality control metric threshold for passing the test established, and a quality control and assurance program established. It is so important to have a quality for all the samples that you're looking to sequence. So, to have a conclusion in this type of presentation, uh, we have to think that the aim of the test is to identify couples in which both parents are carriers and to prevent serious genetic disorders in their offspring. It is so important to think and to pass it through this information to our our colleagues and persons that we know that it is so important that we have this type of test to analyzing to prevent serious genetic disorders. The CarrierSeq expanded carrier screening kit identifies variants in hundreds of genes across hundreds of samples with high analytical sensitivity, specificity, and occurrence accuracy across the CDS and target non-coding regions of 420 genes, including technically challenging targets. The CarrierSeq kit is a research solution that provides all reagents and material for a comprehensive and flexible NGS-based expanded carrier screening workflow. And when you use it with the Ion Gene Studio S5 system and the Carrier Reporter software, the kit enables a simple end-to-end workflow for the detection of carrier-positive samples by research labs interested in maximize the identification of carrier status using genome DNA isolated from blood or saliva samples. I wanted to thank all of you for being here and to see this presentation. And I wanted to thank Thermo Fisher Scientific in the United States and of course the Thermo Fisher here in Brazil. And I'll be able and I'll be joining you guys to uh, our Q&A part right now. If you have any doubts, any questions, I'm more than welcome to reply you all. Thank you so much.
Thank you, Bruno, for your informative presentation. Our first question is, in reference to your slide on analytical validation, what was your analytical validation experience like with CarrierSeq?
Well, thank you so much for your question. I had a really good opportunity to perform the analytical validation, and of course, Thermo Scientific here in Brazil helped me with everything that I could perform to have a good analytical validation here in Brazil. So all the technicians that works in Thermo Scientific helped me to analyze the samples, to choose which samples do we wanted to sequence, to help me to like everything that we need in our kits, our uh, everything that works with a kit. So it's really good about this kit that we can use uh, the kit for every single part. So when we receive the kit in our laboratory, we receive it everything. So we receive the all the parts for library prep to sequencing to make the template in the ion chef. So everything works so smooth that it for me was a really good experience to perform this analytical validation.
Thank you. Our next question is, what were the biggest challenges you faced and how did you resolve them?
Well, this is a really good question. In my opinion, one of the biggest challenge was to really to have the confidence that some challenging genes, as we saw in the presentation, that really worked with this panel. So everyone that works in a lab, we have some these beliefs, and we do think that some genes we must have another type of analysis, that we must have uh, some complemental analysis or performing with another methodology. And for us, was a really good part of the this experience to see that we are able to see those genes. We selected some samples that were positive for those challenging genes, and we were able to see that we found uh, we found the variants. So it was really inspiring for us that we are able to analyze a lot of samples with just one methodology, and this was really good for us.
Thank you. Next question, what is the sample size for NGS based research panel validation?
Well, I can answer with what we performed in our lab to have this validation. So we selected around 100 samples, and of course we do selected the NI12878, and also some samples that we had in our lab to that we have positive controls, that we knew the variants and everything, but we around 100 samples we selected and performed. Of course, some of them were positive, some of them were negative, and we wanted to see if the panel could check everything that we wanted, and we had a really good experience with that.
Great. Well, it looks like we have time for one more question. Can we use cord blood for this experiment?
Well, we experienced since when you have the DNA extraction, you can analyze any type of DNA that you can have. So even a saliva kit, swab, or blood cord, we can use it. You have to have a really good DNA extraction, right? So you have at least around, for our experience, 15 nanograms of DNA per microliter, of course. But when you have a good DNA quality or DNA quantification, you're good to go, and you can have this panel to analyze every single sample of it.
Thank you again, Bruno, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast.